So for today we will see about the protein structure prediction. Okay. So sorry, protein structure determination. Okay, how you can solve the structure that is experimentally. Okay, there are mainly two methods, X-ray crystallography and NMR, that are employed in order to determine the protein structure. So one of the method is X-ray crystallography and NMR. Okay. Now you will see about the X-ray crystallography. So this is the homepage of the protein data bank database. It contains the experimentally determined structure information. So now there is a provision to see the predicted structures also. Okay. And software that is used for protein structure prediction in this particular protein data bank is AlphaFold. Okay. So two methods mainly employed to determine the protein structure, and it's not that much, the experimental procedure is not standard or it is not set. Okay, you have to crystallize or to make the protein in a crystal form. Different experimental conditions has to be followed. Okay, so you have to trial and error. So that process is there in order to set the proper buffer, temperature, in order to get the good crystals, in order to have a proper diffraction pattern. Okay. So more than 1 lakh proteins are available. That is more than, now it's going to reach 2 lakh. Okay, are available within the protein data bank database. So mainly three key steps, that is protein isolation, protein purification, and protein crystallization. These three are the steps that are involved in the process of preparation of crystals. So why we call it as X-ray crystallography is we solve the protein structure using the X-ray, which is having the wavelength of Armstrong unit, which is equivalent to that of atom size. That's why there are many radiations or waves available, but we use X-rays because the wavelength, it matches with the atom's size. Okay, so that's why we are using X-rays. So why crystals used to do X-ray diffraction? Crystals are used because it helps amplify the diffraction signal. Okay, so that's why the diffraction, that is crystals, they used to diffract the rays. Okay, so this diffraction signal actually is the data, electron density, that is captured in order to solve the protein structure. How do the X-rays probe the crystal? So X-rays, they interact with the electrons surrounding the molecule and they reflect. So that reflected, that is the waves, or there is their electronic distribution. Based on that, you get diffraction pattern. So that pattern gives you the information of what is the residue in the particular position, what is the atom in that particular position. Okay, so in that way, you get the complete three-dimensional structure of protein. So what is mainly measured is the electron density. Right. In an atom, based on the electron density only, they diffract the waves, and from that only you get the diffraction pattern. And that pattern is again computationally resolved in order to get the three-dimensional structure of protein. Okay, so this is the main concept behind the X-ray crystallography. So this X-ray crystallography, it provides the finest visualization of protein structure currently available. Okay, so finest and also more accurate. Okay, a single snapshot of a protein in a particular conformation can be obtained using the X-ray crystallography. So this technique can reveal the precise three-dimensional positions of most atoms in a protein molecule. So the use of X-rays provides the best resolution because the wavelength of X-rays is about the same length as that of a covalent bond. So that's the reason. So you have to understand why we are using X-rays in order for the to solve the structure of a protein. Okay, so it's not that only protein structure can be solved. Even nucleic acid structure, RNA structure can also be solved using the X-ray crystallography. So this is a simple picture which gives you an idea how the X-ray beams, they are passed to the crystal. So this is the crystal. When X-ray beam is passed, the electrons in it, they diffract the rays, and the pattern which is collected here, it is detected by the detector. Okay. So this pattern is again computationally resolved using many softwares. Their electron density map is built into a proper three-dimensional structure. So the three components in X-ray crystallographic analysis are a protein crystal, a source of X-rays, and a detector. Okay. So what is a crystal? So whether a crystal will contain only a single monomer of a protein, it will contain many, that is the structures are packed within the crystal in a symmetrical form. Okay, so that's why that is that is have forming a crystal. Okay, so the unit cell is a basic building block of the crystal. So the unit cell can contain multiple copies of the same molecule whose positions are governed by symmetry rules. So the molecule, they are packed in a proper arranged manner, which will give you a crystal. Okay, it's not that when you isolate, when you are going to purify the protein, you will get just a single structure of a protein, a single monomer. It's not like that. Many monomers will form the crystal. So anyhow, when you rotate the crystal in front of the X-rays, the atom they used to diffract. That is, when the same atom is going to diffract the waves, they reinforce each other. So the wave principle is there. That is Bragg's law. Okay, so based on that, they even though many monomers are there, they will have produce a diffraction pattern of a single molecule, which can be used for the accurate determination of the protein structure. So this is the X-ray diffraction apparatus. Okay, here you can see there is a freeze, that is a no, liquid nitrogen. Okay, to make the crystal in a, they should not crack or should not get damaged, right? So liquid nitrogen will pass through it, and when you pass the X-rays on it, the diffraction pattern like this is produced. So liquid nitrogen is used to freeze the control, which allows for increased reliability of information gathered from testing. And the area detector, which collects the refracted X-rays, once they pass through the crystal, is a black plate located behind the nitrogen stream. And sample X-ray diffraction pattern is given here. Okay, like this only you get the diffraction pattern. Okay, so these patterns are used to determine the three-dimensional structure. So the wavelength of the this beam of X-ray, the wavelength of that is 1.55. So this is also one of the important. There has been many objective type questions they may ask. What is the wavelength? It is 1.54 Armstrong. So a narrow beam of X-ray strikes the protein crystal, and part of the beam goes straight through the crystal. And the rest is scattered in various directions. Finally, the scattered or the diffracted X-rays are detected by a X-ray film. Okay, so the blackening of the emulsion being proportional to the intensity of the scattered X-ray beam. So the scattering pattern provides abundant information about the protein. So those electron density which diffracts the X-rays, it forms a pattern. That is, the pattern is the information that is collected in the detector. So that is computationally mapped. The electron, I'll show you that picture at the last, which will tell you the information of how the atoms, the residues are placed in that X-ray electron density map. So the basic physical principles underlying the technique are electrons scatter X-rays. Okay, so that's a principle used here. So the amplitude. So when there is a wave, you have two things. That is amplitude of the wave and also the phase. If you know these two values, you can determine the atomic position. Okay. The amplitude of the wave is scattered by an atom is proportional to its number of electrons. Okay, it's proportional actually. Okay, to the number of electrons within an atom. So the amplitude, it is proportional to the number of electrons in an atom. So that's a carbon atom. How many? That is it's 6 times as strongly as a hydrogen atom. So because the electron in it is 6 in carbon, in hydrogen it is 1. Okay, that's why it is scattering 6 times more than that of a hydrogen. Okay. So the scattered waves, they recombine. So electron atom contributes to each scattered beam. The scattered waves, they reinforce one another at the film or the if they are in phase. And they cancel one another if they are out of phase. So anything, waves, when they cross each other, that is when reinforce each other. So the same thing, property is done here also with X-rays also. Okay, so when the waves, they reinforce on a that is if they are in phase, they reinforce and they cancel if they are out of phase. So the wave in which the scattered waves recombine depends only on the atomic rearrangement. So it's mainly based on the atom arrangement within the molecule. Okay, the way how do they scatter the waves? Okay. So what you have seen in the picture, that is a crystal is mounted and it is positioned in a precise orientation with respect to the X-ray beam and there is the film, detector film. Okay. The crystal is rotated so that the beam can strike crystal from many directions. Okay, so many directions. So we know that when the same atom, that is, if they are phased with each other, they reinforce. Right? So that's why in different directions they rotate the crystal and pass the beam. The rotational motion results in an X-ray photograph consisting of regular array of spots called reflections. Okay, so the intensity of each spot is measured. These intensities and their positions are the basic experimental data of an X-ray crystallographic analysis. Okay, so that's why when you see this diffraction pattern, we can see center it is whole. The other patterns, that is the reflections, they are the information that you collect from the X-ray crystallographic experiment in order to solve the protein structure. So, how you can construct the image of the protein, that is structure of the protein from the detector, that is the film, the image diffraction pattern that you have collected. Okay, so in light microscopy and other microscopies, they have specialized lenses that can give you the idea of an image, right? That's why the principle, that's how the electron microscopy principle works. Whereas the case of X-rays, it's not possible because no appropriate lenses are available to show you the image directly. Okay, that's why there is a need of a mathematical concept called Fourier transformation, which is required in order to that is compute the that is to elucidate the image of the protein. Fourier transformation. So for each spot, okay, this operation yields a wave of electron density whose amplitude is proportional to the square root of the observed intensity of the spot. So each wave also has a phase, that is the time of its crest and troughs related to other waves. Okay, so each atom, based on how do they reflect on the diffraction film, that is the pattern, there will be some crest and trough. That's like a peak. Okay, they only have the question. So based on how the patterns are distributed, they will be having all those things. But determination of this amplitude, that is, you can compute the amplitude, but you cannot know the phase of the X-rays. So that's why they call it as the phase problem. Okay, so the phase of the base is very difficult to determine. So that can be solved in many of the methods, which I will explain you in the next slide. Okay. So each wave also has a phase, that is the timing of its crest and troughs related to those of other waves. The phase of each wave determines whether the wave reinforces or cancels the waves contributed to the other spots. These phases can be deduced from the well understood diffraction patterns produced by electron-dense heavy atom reference markers such as uranium or mercury at specific sites in the protein. Okay, so we don't know in 3D space how the protein, which atom is located in which position. So in order to know that, what you can do is, because you know the sequence, of course we can know the sequence of a protein, but what is its structure is it the task which we have to solve. So in that case, if you don't know how they are positioned, okay, until you know the phase of the particular waves, or the diffraction pattern, it's not possible to determine. So at that time, one thing what you can do is you can place a heavy atom like uranium or mercury. You can, that is, make the protein to bind to a particular uh, heavy atom. So if you know the position where it binds to your protein, then you can use it as an indicator in order to that is understand what is the phase of each of the wave that is distributed. Okay, the pattern. So how the phase problems are solved? So one method is isoformous replacement. So combination of different data from the native crystal with data from other crystals containing the same protein packed in the same way but adding a heavy atom. So you have a crystal with heavy atom and without heavy atom. So like that, if you have many crystals and if you solve the data, you can overlap those electron density map. You can have the accurate structure of the protein. Okay, then that is you can identify, that is determine the calculate the phase of that protein. So molecular replacement, replacement of a known relative structure in different positions and orientations, they provide approximate phases. So you can place the different positions. You can change the structure in different positions, and you can determine the phases. The next is multi-wavelength anomalous dispersion. So different length, there is different variation of intensity distribution in the diffraction pattern over a range of wavelength. Okay, you can just change the wavelength of the rays to some small variations you can make, and you can determine the phases. The next is the direct method, that is the electron density. If you know, have the knowledge of the electron density distributions in crystals, then you can calculate the phases directly from the experimental data. So now the stage is, now you have got the electron density map. That is until you know the amplitude and the phase of that particular electron density map, you cannot have this electron density map in a proper manner. Okay, so once you have, which gives a density of electrons at a large number of regularly spaced points in the crystal. So this three-dimensional electron density distribution, it is represented by a series of parallel sections stacked on top of one another. So each section is a transparent plastic sheet on which the electron density distribution is represented by a contour. So here you can see some of the black lines, right? So this pink, that atoms, that line, it's the position of a particular, that is myoglobin, which is placed on the electron density map. So in diffraction matter, from the diffraction pattern, you get electron density map, right? So that electron density map is these lines. You can see here this iron molecule, how it has the electron density map. For each atoms, there is, you can see the contours, crest, curves, they say, right, in the electron density map. So those things, these represents the electron density map, and when you have the data, only you will have this. This pink color, that here myoglobin, you will not have that information. You have to fix that molecule in on this map in the proper position. So multiple refinements have to be done in order to do that. So this section of the electron density map shows the heme group. The peak of the center of this section corresponds to the position of a iron atom. So when the phase is determined, three-dimensional coordinates, relative mobility of atoms, model built over it, and refinement of the model comparing with empirical data and optimized. You have to do several refinements in order to get a structure in a proper, that is accurate resolution, and also optimized protein structure can be obtained. Okay. So, in which Armstrong, which type of structure can be seen? So, when you have a 6 Armstrong structure, only what is a polypeptide chain, where it is present, so that information you can get. When it is having a 5 Armstrong or 10 Armstrong, the polypeptide chains, how they pack together, so that their centers are between 5 Armstrong and 10 Armstrong apart. So that distance can be measured. So maps at the 2.8 Armstrong and 4 Armstrong, we can delineate the groups of atoms. Okay, so when you have the structure with a resolution of 1.0 and 1.5 Armstrong, you can also map the individual atoms in the electron density map, so which will give you the accurate data. So you can see here the blue contours, right? So those are the electron density maps, and you have to fix the atoms on it, on the map. Like this only, then you require the bioinformatics support in order to solve the structure. After you get the pattern, rest of the things are done computationally, where many softwares like Food and there are many softwares like that which can be used to solve the protein structure. Okay.